the organizers of this great international conference on monocots for the invitation. And I thank you for your time to attending this talk. This is the result or preliminary ongoing results of a collaborative project that involves a large number of people, both students, other researchers, plant collectors, and all sorts of colleagues. So, well, it's a collective effort. This morning we had a talk on Epidendrum, so many aspects of its distinguishing features and overall characteristics have been already explained by Eric Hágsater. I only would like to stress a few aspects that make it very interesting for an evolutionary biologist like myself. One, it's huge size. I think Epidendrum is competing now with Piper of the Piperaceae for the title of the largest genus of plants in the Neotropics. And with the present data, surely it appears to be the largest Neotropical orchid genus. It is found throughout the Neotropical region and even a bit out of the tropics in the north, in Florida and southeastern United States. But also, this taxic diversity is mirrored by a huge disparity of ways of living. Eric explained to us how the plants can be hanging or erect, having pseudobulbs or thin stems. And this plasticity enables them to occupy almost any niche available for plant life in our continent. And from sea level mangroves, such as Epidendrum nocturnum, up to over 4,000 meters elevation in the Andes in the case of this species. Well, Eric already told us of the differences in sizes and structures of the plants. Sorry, it's me. And also explained us some of the unique features of the genus. And I am very impressed by the fact that even if Epidendrum displays all sorts of sizes and odors and floral forms, the basic structural organization is very conserved. All of them have this keyhole floral structure with the fusion of the column to the base of the labellum, which generates a tunnel. In some instances can be very long or very short or very wide. And in some instances, we have found a nectar. In others, there's no obvious reward. But the point is that apparently this very canalized floral structure appears to be very good for making new species. I suspect that much of the high taxonomic diversity we find in the genus perhaps has to do with the specialization for different pollinators. But it would be nice to have more data on pollination. There are very few studies focused on that. Trying to find order in this huge genus, Eric Hágsater and co-workers have studied the taxonomy of the genus for several decades. Eric Hágsater has proposed a number of informal morphology-based groups. And it is interesting that when he and collaborators started to testing the existence of these groups using an independent source of evidence, in this case, DNA sequences of one plastid and one nuclear DNA regions, many of the morphological groups were apparently confirmed. And in contrast with that apparent congruence between morphology and DNA, we found very little support for most of the relationships they recovered. This is a combined tree, parsimony tree based on nuclear ITS and plastid gene MADK. And you can see that the Epidendrum Alliance has some bootstrap support, 73 or something like that. A few of the terminal clades have some support, but the spine is essentially unsupported. Subsequent studies have explored the effect of adding more information. For instance, this work by Hágsater and collaborators included several plastid markers, intergenic spacers, coding regions, and the nuclear ITS region. And for the particular case of the Epidendrum anisatum group, well, there was some improvement. Several of the relationships attained a higher bootstrap support, but there are still many parts of the tree that are unsupported. In recent decades, the lower costs and higher speed of generating large volumes of genomic data has opened the opportunity to approach these old questions with new tools. And a few years ago, we had a project, an exploratory pilot project, investigating the usefulness of a technique that had been applied to several animal groups. It was starting to be applied to plant groups. It was one variant of target sequencing called the AHE, or hybrid anchor hybrid enrichment, developed by these people here. And it consisted in designing probes for capturing and later enriching and then sequencing about 512 nuclear genes of low copy nuclear genes or single copy nuclear genes. And we also, from the same data derived from that sequencing, we're able to obtain essentially all the protein coding plastid genes. So when we analyze the data, well, for a start, we wanted to know if that technique would work for Epidendrum. And we found that many of the species recovered a high number of loci. For instance, all the species had at least 200 out of the 500 target genes sequenced. The length of the reads varied from 160 to 1,500 base pairs in length, and with an average of, I don't know, say 600 and something. Anyway, we performed an analysis of phylogenetic informativity and found that, well, most of the nuclear, the coding loci that we sequenced informed around here. This is an arbitrary time scale, the present and the root of the clade we are analyzing. And we see that most of the information generated by the coding nuclear genes is near the present or shallow phylogenetic relationships. The plastid markers usually are evolving more slowly and inform a deeper evolutionary time. And there was a surprising, well, not so surprising because somebody else had already noticed the high phylogenetic informativeness of this marker, the gene YCF1, which is very long and the most informative plastid marker so far, and it was even more informative than most of the nuclear markers we sequence. This is a comparison of the trees resulting from the nuclear data for that sample of species and the plastid protein coding genes. And as you may notice, well, for instance, the support for the clades increased substantially. Remember, please, that the spine had no support with the individual Sanger sequenced markers. Look at these values. And the overall relationships are congruent, and the few incongruences are parts of the tree for which there is very little information provided by these markers. So essentially, they are telling us more or less the same story. Then we decided to continue with this approach. And earlier I mentioned that when we got funding and started this new part of our project, we had to make a decision about what tools we would use. Since we already had promising results from this technique, and given that the results for Asparagales attained by the angiosperms 353 set of probes from Q didn't look very promising, we decided to keep doing the same markers, the same strategy, but we enriched the set of probes to target about 700 loci, including several which are interesting for us because of their ecological or physiological function. For instance, genes related to CAN metabolism or the control of floral color and the like. Anyway, and we wanted to produce a strong, a solid, robust phylogeny as a framework for answering a number of questions. For instance, to produce an objective, informative, predictive classification, and also as a basis for further macroevolutionary studies. And we are interested in particular aspects such as morphological evolution and evolution and the evolution of the size of the nuclear genome. Well, then we did this Hyb-Seq approach, target nuclear genes. We modified our original strategy for enriching only 70% of our extract, and 30% of the extract was not enriched for the nuclear genes, so did random sequencing for that portion, and we were able to increase the production of reads from the chloroplast genome and the mitochondrial genome, and then we have access to all three genomic compartments. As preliminary results, we were able to assemble a number of new plastoms for Leline. Our main focus, of course, is Epidendrum, but we also sequenced most, if not all, of the species of, or nearly all the species of Barkeria and Laelia, and about 15 or 20 additional genera of Leline for comparative evolutionary purposes. The structure of the plastid is the standard one with 4 partitions, and there was very little variation in size. The smallest plastid genome was this size, and the largest was this size. When comparing closely related species, for instance, all the species of Epidendrum, we found very little variation in the structure of the plastid genome, as in the case of Barkeria. In this case, we see some variation in the length or the limits or the order of some genes, but please notice that those exceptions belong to different subtypes. Overall, the evolution of the plastid genome in Epidendrum seems to be very conserved. Now, we were able to build preliminary phylogenies using the whole genome plastom. We made an effort to check the alignments for the non-coding regions, which is not necessarily trivial. And then we obtained very well resolved trees with relatively high support for many of the clades. And we started using these trees to ask questions about different comparative aspects. For instance, here, I think I'm doing something wrong. Here we mapped or we are showing the presence or absence or reduction of the NHD gene family. We use members of other subtribes and tribes as outgroups. And please notice that in all of the outgroups, the whole family of 11 NDH genes are present. But within Leline, which starts here with the blue color, and in Epidendrum, there are large losses of this family of genes. In some instances, well, when you see color, the symbol, it's that the gene is present and apparently functional. I don't see, well, there are some other instances where the square is empty. It is not obvious here. That means that apparently it was pseudogene, it is a pseudogene. And in many other instances, they are simply lost. And we found some interesting patterns, which we cannot explain yet, for particular clades that apparently recovered or didn't lose some genes of this family. But these are questions that we have to pursue further. Now, I am presenting here a tree based on the nuclear single copy or low copy genes. We also had a very good resolution, and this is this part, the sister clade, which consists of association of clades, including Myrmecophila, Microepidendrum, Barkeria, and the sister group of Epidendrum appears to be this couple of genera, Orleanesia and Caulartron, and Epidendrum is strongly supported as monophyletic, and many of the relationships within the genus are much better than in prior analysis. One thing that is very important to notice is that when we compare, well, I'll go back to that quickly later, but I want to show you some examples of particular clades that were delimited at first from morphology, and then when corroborated by ITS without support, and we are finding them with much better support here. That means that taxonomists are doing a good job with the tools they have at hand. And a question I want to address here is how similar or different are the relationships provided by the T3 genome compartments. And to my pleasure, most of the relationships are congruent with particular cases of different positions among the mitochondrial, plastid, and nuclear markers. But these are particular examples or particular cases that we want to examine in more detail, because as you know, there are a whole set of probably causes for incongruence, and this is not a problem any longer. It's part of life. No? Each gene tells us its story. So I grew up with the idea in the early days of my phylogenetic studies that incongruence was a problem. No, it's a fact of life. We have to explain it, not to avoid it or put it or hide it. Well, just two further questions. What are we using these trees for? For instance, we are trying to sort out these taxa that the taxonomists didn't know where to put. Now we are having confirmation that morphology, floral morphology is a good predictor of phylogenetic relationships in this case. And now we can place or recognize relationships we didn't suspect earlier. And we also are having information to ask interesting questions, such as the fact that, as we already said, Epidendrum is characterized by the fusion of the labellum and the column. There are derived clades that have a complete separation of those two organs. And if you see this image and compare it with this one, please notice that even though they are closely related, this one and these ones, the separation of the column is accompanied by other modifications, such as a simplification of the perianth. So there are very interesting phenomena to be investigated there, such as, are these changes pleiotropic? I mean, it's a locus that has effect on several aspects of the morphology. We don't know, but that's a line that we would like to pursue further. I'm going to skip this one. I just want to acknowledge and thank my group of collaborators, my brilliant students, and all the people who have contributed to this work. Thank you.
We have time for some questions. Any?
Nice talk, Gerardo. I was just wondering how to deal with these incongruences for the classification that we are trying to. We know you are trying to. You are trying to produce, like maybe take out all the incongruence and then mix them, and then I don't know how to. What you think about it?
I think we have to go to those particular incongruent species and look at them with more detail and trying to find out what is happening. For instance, in some instances, we have noticed that the recovery success for the nuclear markers was smaller. So it may be that there are many missing data that are confounding the pattern. In other instances, it could be an ancient hybridization, or there are several possible causes. So we are at the phase of noticing the situation, and now we are in a position to explore it more deeply and try to find explanations. But at this point, we don't have them, only hypotheses to test.
Another question? No? Thank you very much.
Thank you.